Hello, now I'll be sharing our work on D-Carbamoylases, enzymes that play a key role in what's known as the hydrogenase process. This process is used to produce optically pure D-amino acids, which are crucial components in the pharmaceutical and food industry. What makes the hydrogenase process particularly attractive is its environmental friendliness and safety. Compared to traditional chemical methods, this biocatalytic method is non-toxic and sustainable. Two enzymes are involved in this process. First, dehydrogenase, which opens the hydrogen ring structure, and then D-carbamoylase, which removes the carbamoyl group to generate the final D-amino acid product. Together, they offer a greener alternative for production these valuable compounds. In this project, we set three main goals. First, we wanted to characterize a D-carbamoylase from ensifer adherence F5. For this, we used a combination of computational modeling, tracking studies, and molecular dynamic simulation to get a detailed picture of how the enzyme behaves and interacts with substrates. Second, we moved from in silico to the lab. We expressed the enzyme in E. coli host and analyzed its properties. And third, we compared the enzyme with previously studied D-carbamoylase from Pseudomonas species. This dual approach, computational and experimental, allowed us to gain a comprehensive understanding of the enzyme. The main methods that we used are listed on the slide. We began by analyzing publicly available amino acid sequences of E-adherence D-carbamoylase and predicting its 3D structure using AlphaFold 2. The quality of the predicted model was excellent. The PLDDT confidence scores mostly above 90 and in many regions close to 100. Once we had the structure, we moved to the molecular docking. We tested the binding of 5 different N-carbamoyl D amino acids. The model showed strong affinity across all ligands, with a particularly high preference for N-carbamoyl D tryptophan. Interestingly, the results for N-carbamoyl D tryptophan mirrored our earlier findings with the Pseudomonas enzyme. To dig deeper, we ran 100 nanosecond molecular dynamic simulation on both unbound enzyme and the enzyme ligand complex. The RMSD and RMSF analysis showed that the structure were stable throughout the simulation and the enzyme maintained acceptable fluctuations of amino acids, nothing unexpected. We also looked at the radius of gyration to understand structural compactness. In the case of enzyme-ligand complex, we saw some initial dispersion during the first 10 nanoseconds, but then both the free and bound forms reached a similar level of compactness. Notably, the E-adherence enzyme stabilized faster than the Pseudomonas enzyme. We checked solvent accessibility as well using SASA and found little difference between the bound and unbound enzymes. This suggested that ligand binding did not significantly impact the enzyme's exposure to solvent. It was important that the enzyme formed up to 6 hydrogen bonds per nanosecond, with N-carbamoyl D-tryptophan indicating a strong and stable interaction. The total binding energy was -15, which supports the docking data. Finally, we analyzed the free energy landscape. This showed that the free enzyme samples a wider range of conformations, while the enzyme-ligand complex adopts a more compact and stable structure, consistent with ligand-induced stabilization. After the promising computational results, we moved to the lab to test our predictions. We synthesized the gene of the E-adherence D-Carbamoylase and expressed it in E. coli. However, we faced a common issue. Most of the protein accumulated in the insoluble fraction. To recover it, we use N-lauryl sarcosine sodium salt, a mild detergent, to solubilize the protein for analysis. SDS page confirmed the expected subunit size of 39 kilodalton, and the tetrameric form of the enzyme was about 148 kilodalton. Next, we performed enzyme activity assays using the same N-carbamoyl D-amino acid substrate from our docking study. As predicted, the enzyme showed clear activity against these substrates, but no activity against L forms, confirming this stereospecificity. Moreover, it was active toward both aromatic and aliphatic substrate, which nicely aligned with our in silico findings. In this study, we compared D-carbamoylases from E-adherents and Pseudomonas, observing distinct substrate preferences and catalytic behaviors. While Pseudomonas enzyme showed higher overall efficacy, the E-adherence enzyme remains promising. Improving its expression or solubility could significantly enhance its performance. With further engineering, it could serve as an effective biocatalyst for D-amino acid production. Thank you very much for your attention. I would be happy to take any questions.